Hi, welcome to this brief lecture on genetic heterozygosity. Now, before we dig into the genetics of this topic, I really wanted to make sure you guys understood what I'm talking about when I say a phenotype. So organisms often have a phenotype that they express, and that phenotype can have a range of features. For instance, these two dogs in this example, one dog has a phenotype of being very tall, being huge, and the other dog is kind of a dwarf. And so we'll come back to this example, but I want you to have in the back of your head that there is a continuum of phenotypes within a species of organisms. So to just kind of orientate you guys, I want to talk today about what is heterozygosity and why it's important for us in human biology. And then there are a few sort of major learning outcomes I want you to keep in the back of your head during the lecture. Many organisms are diploid, which means they have two copies of every chromosome, and slight variations of genes within one of those chromosomes can lead to one gene being dominant over the other. And if a diploid organism contains both a dominant and a recessive gene, then we call that a heterozygote. And heterozygosity is critical to the genetic diversity of organisms, which leads to the phenotypic diversity that they show, right? Just like those really tall and dwarf dogs. So hopefully you all remember that DNA is the blueprint for the diversity of life, and it is packaged in the cells. It's really tightly wound into paired chromosomes, which you can see at the bottom of this diagram. And this is two chromosomes that are linked here at the centromere. And next to it is a fluorescent image, micrograph, of paired chromosomes in a human cell. And so mammals are diploid organisms. And that's critical to understanding the interplay between the genes, because each chromosome has the same gene on it, and those two genes are interacting with each other to be expressed. The gene that is favored, that is always expressed, is called dominant, and the gene that is not favored is called recessive. And a heterozygote contains one each of a dominant and a recessive gene. And so we can kind of use our knowledge of the offspring frequency of heterozygosity and dominance and recessive in a Punnett square to understand phenotypic frequency that we would expect. And so in this top Punnett square, you can see there's capital A represents the dominant allele in this gene, and lowercase a represents the recessive allele. And when we do crosses, we get a quarter of our offspring are homozygous dominant, and then a quarter are heterozygous, and another quarter are heterozygous, and then another quarter are homozygous recessive. And because this gene to being tall is dominant, that means that 3/4 of our squares all express the tall trait, and only 1/4 expresses the dwarf. But there is a range of phenotypes in this population. Whereas if in the population, if all your parents were homozygous dominant, then all you would ever get is 100% tall offspring. And the same thing is if your parents were all homozygous recessive, you would only ever get dwarf dogs. And so critically, having heterozygosity in a population allows for phenotypic variation that can respond to selection and stress. And we can sort of see this in human diseases. Sickle cell anemia is a really terrible disease, which is recessive, in which a gene has a mutation that destroys the natural shape and function of red blood cells. And so when the cells sickle like this, they can no longer hold oxygen, and they can actually cause a lot of pain. And only offspring that are homozygous recessive will express sickle cell anemia. But the reason this disease, even though it's very debilitating, persists in our population is because there are heterozygotes that are carriers of the disease, and they just don't express that disease. And this is a really critical component of our genetic testing. When we test human embryos, we're checking the parents to see if they're heterozygotes for a particular disease, and if there's a chance that they will produce an offspring that is homozygous and actually express that disease. So while diseases are terrible for humans, I would argue that heterozygosity is a really critical feature of genetic and phenotypic diversity. Thank you.